Well, thank you very much for coming today as we sort of start our summer holidays, I guess. Yes. Telling Adam, but if it came in August, we'd probably only have 1 or 2 people. But anyway, so it's really a pleasure to have Adam. It's kind of our ongoing connection with Monash University. As you know, Erica Sloan shares a joint appointment in the Cousins Center, also in Monash University. And they introduced us to Adam, who's doing this really great work looking at sleep and circadian rhythms in relationships to neuroinflammation and chemobrain. He is a National Breast Cancer Fellow in the Neuroregulation Laboratory at Monash University, which is in Melbourne. We don't know. He earned his PhD at the University of Newcastle in Australia as well, and then migrated for a postdoc with Robert Dancer at MD Anderson in Texas. He's been examining the biological mechanisms that cause memory and learning impairment and mood disorders in cancer patients. So primarily on animal models. He's also identified, I think really interestingly, that solid tumor, just the presence of a tumor, produces changes in cognitive functioning, and is understanding to what extent that may be mediated by neuroinflammatory processes. In talking with him today, I really learned about the importance of our daily aspirin use, that it can actually have impacts upon cognitive function. So I'm looking forward to seeing some of those data and hearing about other treatment methods that he's using to address chemobrain issues in breast cancer survivor populations. Welcome very much, Adam.
Thank you very much for the opportunity to share some of the work we've been doing back in Melbourne. I realise that I haven't Americanised my slides, so you're going to have to put up with the Queen's English in terms of spelling today. I'm not actually going to talk much about sleep and circadian rhythm work that we're doing, but I definitely will be talking about neuroinflammation and aspirin, and I'm not getting paid by Bayer to tell you about the benefits of aspirin, I promise. So it goes without saying that cancer treatment is absolutely necessary for cancer survival, and is becoming increasingly successful. If you look, at least for a number of cancers, if you look at breast cancer, for example, unless you have metastatic disease or triple negative breast cancer, you stand at around a 90% chance of survival. Colorectal cancer is the other cancer that I'm particularly interested in, and you stand at around 80% chance of survival for that. However, treatment does come at a cost. And in fact, over 70% of cancer patients will report problems with cognition and mood symptomology, such as depression or learning and memory, executive function, concentration difficulties. And we know in a subset of patients that this is chronic. It's been reported at least up to 20 years post-treatment. Now, we currently have no treatment for cancer-associated cognitive impairment, and I would argue that's because we don't really understand the mechanisms, and perhaps we don't fully appreciate the mechanisms because we've been quite singular in our approach to studying the insults of treatment that cancer patients will receive. In the past, we've typically focused on the effect of chemotherapy, but there are a range of events throughout a patient's cancer career that we know are able to induce damage to the brain and impair function. So we hypothesize that as a patient moves through their trajectory of treatment, there can be an exacerbated effect from diagnosis through surgery. 80% of cancer patients with solid tumours will undergo surgery at some point. Adjuvant therapies such as chemo, radiation, hormonal therapies. And we believe that there's going to be a consistent increase in exacerbation or prolongation of side effects as these events are added on top of each other. Now, there are some patients who do return back to baseline functioning or close to, which we would consider to be somewhat resilient, but there are a subset of those patients, as I mentioned, that really transition from the acute effects of cancer treatment into chronic cognitive and mood dysfunction. So we want to explore the biological mechanisms that might be causing these effects, and we've chosen to do that by translating backwards into mouse models. Now, we can't do everything in a mouse model, but we can start to try and add a few key events that we're interested in to see if there is this cumulative effect. Excuse me. So we're particularly interested in looking at the combination of the cancer itself, surgery, and then adjuvant therapies such as chemotherapies, to determine whether or not we can see this separation at some point in regards to resilient mice versus susceptible mice, and then dissect those mechanisms in between. So far, we have focused on the effects of these key treatment events individually, because if we want to know if there is an exacerbation when these events are placed in combination, we first must characterize what's happening individually. So I'm going to spend the majority of my time today talking about cancer-induced cognitive impairment and mood dysfunction. There's evidence clinically to indicate that that patients at baseline, so prior to any treatment whatsoever, present with cognitive impairment. So this is some work from some colleagues of ours at the University of Sydney, in which they've showed in a large-scale prospective clinical study of colorectal cancer patients that colorectal cancer patients present with increased incidence of cognitive impairment using objective neuropsychological test batteries compared to age-matched healthy controls. You'll notice that the incidence of cognitive impairment does not actually dramatically rise even after these patients receive chemotherapy. So there's something that's happening to the patients at baseline before treatment. So many have argued that it's the stress and anxiety of diagnosis that may be contributing to this, and no doubt it does. However, we've started to ask the question, well, perhaps it's the cancer itself, a peripheral solid tumour that can hijack the brain. This is a question that's really, really hard to dissociate in patients. I think you can't really ask a patient not to be anxious or stressful once you've gotten a cancer diagnosis, but we can do this really easily in mice. I just don't sit them down and give them their diagnosis. We just put the tumour cells in and see what happens, right? So the aim of the first half of today's talk is really for us to examine the role of the tumour in cognitive impairment. And we've been running with the hypothesis that a tumour is sufficient to induce a neuroinflammatory response, and this leads to cognitive impairment. So how do we go about doing this? Well, we use a syngeneic orthotopic mouse model of breast cancer. This particular one is a 41.2 breast cancer cell line. Control mice will just receive the vehicle, which is PBS. So we inject that into the fourth memory fat pad of mice. I'm sure if you know Erica's work, you're familiar with the animal cancer models that we use. These cells are tagged with luciferase, which means that we can do bioluminescence imaging to track in real time in vivo the tumour progression metastasis in our mice. We also examine sickness responses throughout the experiment. I'm going to present data where we've investigated the effect of these tumours on hippocampal dependent episodic memory in our mice, and of course we collect tissue for biological or biochemical analyses. So how do we measure memory in a mouse? Well, one way is to do the novel object recognition task, and this is where we place the mouse into an arena and we give them two identical objects to explore. Mice are curious and they like to explore, so they spend 5 minutes engaging with these two identical toys, and then we take them out for 5 minutes. We give them a break and then reintroduce them to the arena. This time we replace one of those objects with a novel object, something the mouse hasn't seen before. Because they're inquisitive and they like to explore, they should be spending more time engaging with that novel object than the old object. But of course, in order to do that, they have to remember that that old object is old. So this is what it looks like in our behavioural arena. This is after 5 minutes in the test, and the blue lines represent where the mouse has travelled You'll notice in our non-tumour bearing mouse here, she spent a lot more time around that novel object compared to the old object, where it's roughly 50-50 with that tumour bearing mouse. If we quantify that, what we see is that as early as 4 days after implanting tumour cells into the body, mice are already showing significantly less time engaging with that novel object compared to non-tumour bearing control mice. We can look at longer term episodic memory, and this is essentially the same test, but we give the mouse a friend to play with, and then we take them out for 80 minutes this time and we put them back in and give them the old friend and a new friend. It's the same deal. As early as day 3, we start to see tumour-induced reductions in episodic memory. That seems awfully early, right?
So
Sure.
Can I just ask a clarification question? So you've interpreted that as a memory difficulty as opposed to novel seeking or the interest in novel objects. So how do you infer that it's memory and not interest in novel or rewarding, anything that would be rewarding?
So in terms of sucrose preference and anhedonia behaviour, so reward seeking, we don't see any changes there in this model. Also, we see no differences in the learning phase, so the acquisition phase, and at that point, both of those objects are new and novel. So the inference is that it's memory.
How about just activity, baseline activity? Can you see that as being changed?
I'm going to get there.
Okay.
I love that question. I like ones I can answer. So it is seems awfully early, right? So we went in and checked that we weren't actually measuring luciferase-induced cognitive impairment. The tumour cells are syngeneic, but they are tagged with luciferase, and that might mean that there's enough foreign substance in the body to provoke an immune response, a little bit of inflammation, and that's causing the memory change. So we included a third group, and that is mice that receive tumour cells that are untagged. We see exactly the same effect. There's no difference between the two. This is really tumour-induced cognitive impairment. So you might just be asking, are the mice sick, right? If they're sick, they're not running around, they're not engaging in the behavioural tasks. In order for me to answer that, I think it's important that you understand a little bit of the kinetics of this cancer model. From around day 15, if you look at tumour volume by caliper measurement, day 15, 18, that's when we really start to see primary tumour growth take off, which is actually matched by around the time, as you'd expect, where we start to see metastases emerge, and then really tumour burden dramatically increases. If you look at sickness responses such as body weight loss, it's only at that late phase that we start to see reductions in metastasis. But we can also look at a more sensitive measure of sickness such as burrowing. This is how much mice will displace pellets from a tube. They love to do it. It involves a little bit of motivation aspects as well, and it's a sensitive activity measure. It's only when we have high levels of cancer burden do we start, from about day 22, do we start to see this general drop in burrowing, not when I've shown you these changes in memory performance. Of course, there's likely to be memory impairment here, we just can't dissociate that from the sickness. So what is it about tumour cells that are talking to the brain? Well, we asked, well, is it possible that it is something secreted by those tumour cells that interact with the periphery, and that signal propagates to the brain to induce memory impairment? The obvious first question is it these secreted factors? So to test that, we grew up our tumour cells in dishes. The control dishes had no tumour cells, but we put the media in and it goes through the same process. We incubate the cells in media, and then we take that media and filter out all of the cells. We freeze it down, and then we go in and we gave our mice daily intraperitoneal injections of tumour-conditioned media or the control media. We did that for about a week, and then we tested memory function. Well, what we found was that by just injecting those factors secreted by the tumour, we were able to recapitulate tumour-induced cognitive impairment. So, it appears that the tumour releases factors that eventually, or at least which contribute to this tumour-induced cognitive impairments, but we've got some gaps in between here, right? So we asked, what are those secreted factors? And as I mentioned, we're interested in the potential role of inflammation. So we ran multiplex analyses on our tumour cell conditioned media. There are a vast array of cytokines and chemokines that are released by our tumour cells, but you'll notice that there are 4 major candidates, at least in this model, that are increased or released in greatest magnitude. So we asked ourselves, does this inflammatory signal look like the inflammatory signature, in the periphery? And what about in the brain, and specifically the hippocampus that's going to be doing most of the work in our behavioural task? So here we have our tumour cell conditioned media again, and you'll notice that the plasma of our tumour bearing mice show almost an identical inflammatory signature. This model also produces a great deal of splenomegaly. We have a lot of extramedullary myeloid cell expansion. So this would tell us that the spleen is a peripheral target in which we should maybe explore a little further. And it also explains why we have such high levels of GCSF as well. Well, we looked at some candidate genes in the spleen and found that there were significant increases in some pro-inflammatory cytokines and reductions in anti-inflammatory IL-10, which is exactly what we see when we look in the hippocampus of our tumour-bearing mice. Increases in those pro-inflammatory cytokines, decreases in IL-10. We're actually currently running multiplex to check protein on these as well. Okay, so it looks like a tumour secretes factors, cytokines and chemokines. That inflammatory signature is, we believe, reflected in the periphery and in the brain, but we don't know if inflammation is causal in this effect. So to begin to ask whether or not inflammation might be causal in tumour-induced cognitive impairment, and to see if we might be able to start repurposing currently available safe and cheap anti-inflammatories that can be rapidly translated to the clinic, we gave our mice aspirin. So what we did is we went back to our tumour model, we put them, we gave them either tumour cells or the control condition, but we started them on low dose aspirin in drinking water about 1 to 2 days prior to the tumour cell injection, and we kept them on there. PK/PD analyses would tell us that the dose of aspirin we gave them is equivalent to about 100 milligrams per day in a person, so low dose. Aspirin completely blocked tumour-induced cognitive impairment or memory impairment on our novel object task. Just to prove to you that it's not because aspirin reduced our tumours, which is possible, but at this dose we saw no changes in primary tumour volume or growth, at least in this early first week, no changes in activity. So, peripheral solid tumours excuse me, induce cognitive impairment. We believe, I understand aspirin has off-target effects other than inflammation, and we're chasing that down as well. Inflammation, we believe, is causal if the effects of aspirin are on tumour-induced cognitive impairment are occurring via its anti-inflammatory properties, and we can prevent tumour-induced cognitive impairment. However, patients are going to come to the clinic and present with cancer already, which means they could already have neuroinflammation and cognitive impairment. So the real question is, can we reverse the effect? Can we use aspirin or anti-inflammatories potential to treat tumour-induced cognitive impairment? So this is work done by a PhD student of ours. It's really early days yet, so take it with a grain of salt. We've only got about 4 mice per group, which is really small. But what Nijun has done is she's gone in and she baseline tested our mice. So these are untreated at baseline, but they had the same cognitive performance. And then we gave them tumour cells, or one group, half of them tumour cells, to be able to see a drop in their function. So on day 5 we see tumour-induced cognitive impairment. Then Nujung came in and started giving them aspirin to see if we can then reverse that effect. So it doesn't look like we can. Like I said, it's early days. There's no statistical significance, but you can see a trend here towards a drop in both tumour-bearing mice with or without aspirin. So that's a problem, right? It appears that anti-inflammatories may not be able to reverse tumour-induced cognitive impairment. So perhaps we have to look downstream of neuroinflammation. Inflammation might be the trigger, but it's not necessarily the bullet. So I want to take a step back now and talk about what insights we might be able to gain from other models of neuroinflammation-induced cognitive impairment and depression. I'm going to talk quite a bit about depression in a moment, that we can then apply in the cancer context. So this is work that I did at the University of Illinois and at MD Anderson Cancer Center, predominantly with Robert Dancer, in which we were examining the kynurenine pathway in its causal relationship for LPS-induced depression. So now we're talking about a bacterial mimetic, but hopefully we can gain insight to then come back to the cancer context and start investigating that.
Do you mind if I ask you just one question about the previous model first? In terms of the tumor burden in your mouse model in a versus a breast cancer patient, you got a teeny tiny little tumor in most women with early stage breast cancer. How does tumor size factor in? If you think that these are tumors that are
So that's a great question, and I both think it does and it doesn't matter. So we've certainly looked into that. It would seem that the effect of a really tiny tumour, so not even palpable, like in the first week of the mice, is sufficient to cause these memory impairments, and we don't see it getting necessarily worse. But it is an acute mouse model, and the behavioural assessment is not as sensitive, say, as what you could do in a patient and really dissect what cognitive processes are affected versus those that aren't. So it might, but it might not. In the very least, you have a few tumour cells in your body, it's already starting to talk to the brain. So if we move back and talk about this kynurenine pathway, I want to give you the big picture because it gets a little complicated, and we'll walk through the different components. So we believe that once we have a peripheral activation of innate immunity, we have peripheral inflammatory cytokines. So I'm going to talk about that in the context of lipopolysaccharide induced, so LPS induced, but the aim is, or the hope is, that the same goes, or the same story is with cancer. And we certainly see cytokines with cancer. That inflammatory signal in the periphery propagates to the brain to cause neuroinflammation and activation of microglia. At the same time, in the periphery, it activates this enzyme called IDO, indolamine 2,3-dioxygenase, and it converts tryptophan into kynurenine. I'll walk you through that pathway in a moment. Kynurenine also then travels into the brain. It gets taken up by activated microglia and then metabolized further into a range of metabolites that become neurotoxic, and they activate the NMDA receptor, such as quinolinic acid. At the same time, activated microglia are dumping out a whole bunch of glutamate into the extracellular space. They do that in order to exchange it with cysteine, and that maintains the redox status of the cell and basically helps the cell stay alive. So we believe that a combination of excess glutamate in the brain combined with the production of these neurotoxic metabolites that agonize the NMDA receptor leads to depression or cognitive impairment. I'm going to talk about how we can leverage these pathways, both at the level of the enzyme, pharmacologically, but we've also done it genetically. We can actually, for the first time, we have evidence that the blood-brain barrier may be an antidepressant target, and we can stop kynurenine from entering the brain to to begin with. And I'll talk about how we can go downstream and use ketamine to block the NMDA receptor. So this is the kynurenine pathway. Here we have, in response to cytokines, this enzyme IDO is going to metabolize tryptophan into kynurenine. In the periphery, we see a drop in tryptophan, but this is actually in the brains of LPS-treated mice 24 hours later. We don't see a drop in tryptophan there. I have a whole bunch of reasons why, and I can explain it, but in the interest of time, you can ask me questions about it afterwards, but trust me. But this is really important because we originally thought, well, if IDO metabolizes tryptophan into kynurenine, it's not going to be available to produce serotonin. Lack of serotonin is a depression story, and then it's all over. But actually we have an increase in serotonergic turnover. So this isn't a depression and serotonin story. We see no changes in the kynurenine aminotransferase pathway. This is one that produces a metabolite considered to be largely neuroprotective. But we do see a metabolism of this kynurenine down the KMO, or kynurenine monooxygenase pathway, that results in neurotoxic metabolites, quinolinic acid, and activate the NMDA receptor. So we asked, can we intervene at the level of IDO right up here to block this event from occurring? So we use knockout mice, but we can do it pharmacologically as well, in which we tested mice treated with LPS or with control or vehicle on depression-like behavioural tests. Sucrose preference test is a so-called measure of anhedonia. The forced swim test is a measure of learned helplessness for depression. You can take whether or not that's true of a mouse to a human with a grain of salt, but the point is these are tests that are sensitive to antidepressants. So we wait 24 hours in order to let the sickness behaviours of our mice, so it's the other way around this time, subside. So you give the mouse LPS, they act sick, they don't run around. 24 hours, that sickness behaviour is no longer there, but we have a window of opportunity to then measure depression-like behaviour in the absence of sickness. What we found was that wild-type mice showed this drop in sucrose preference, so a depression-like phenotype in response to LPS, but the IDO knockout mice show no evidence of LPS-induced depression-like behaviour. Same goes with the forced swim test immobility. There's an increase in the immobility, which is the indicator of depression or depression-like behaviour. No such increase in IDO knockout mice. As you'd expect, if we look at the kynurenine to tryptophan ratios in the blood, so if tryptophan's being metabolized into kynurenine, we should be seeing kynurenine go up, tryptophan go down, so the ratio should be increased. And that's exactly what we see here in our wild-type mice, but not in response to the IDO knockout mice. There's been quite a lot of work published on using IDO inhibitors as well, so pharmacological evidence to support this. So we can block upstream at the IDO, level of IDO. We can. What about if we go all the way downstream and give our mice ketamine and block the NMDA receptor? Well, I'm sure you're aware of the research showing the beneficial effects of ketamine for depression. We were the first to show that it works in inflammation-induced depression. Again, we see this drop in sucrose preference in our LPS-treated mice. Pre-treatment with ketamine, so an injection of ketamine immediately before the LPS, completely blocks a depression-like behaviour. The same goes on the forced swim test. So as I mentioned, those activated microglial cells are dumping out glutamate, to exchange for cysteine and stay alive. The question is, if we're blocking the NMDA receptor, where's all that excess glutamate going that's in the brain? Well, we hypothesize that since we've shut this down, that glutamatergic neurotransmission is being redirected towards the AMPA receptor. There's a wealth of evidence showing that activation of the AMPA receptor induces antidepressant properties. So we went in and we gave our mice LPS, half of them we gave ketamine, and then half of those we gave with NBQX, an AMPA receptor antagonist. what we found was that we were able to restore on the sucrose preference test that depression-like phenotype. Okay, so it seems that we can target the enzyme IDO and shut things down. It seems that we can go all the way downstream to the effects of the NMDA receptor. What about trying to stop kynurenine getting into the brain in the first place? And this is what I'm quite excited about. We've just gotten reviews back in Molecular Psychiatry. They seem quite promising because this is the first time we've been able to show that the blood-brain barrier is a target for as a treatment for depression, and we may be able to do it with something as simple and safe as a branched-chain amino acid supplements. So what happens? So LPS is going to induce inflammation and turn on IDO. Metabolize tryptophan into kynurenine, and that kynurenine gets into the brain. So we know that the vast majority of the central pools of kynurenine are derived from the periphery. It's 78% at baseline conditions, 98% under conditions of inflammation. Once that kynurenine gets into the brain, that KMO is going to be activated and turn it into the nasty stuff that's leading to depression. We looked at whether or not we can competitively inhibit transport of that kynurenine with leucine. Why? Kynurenine, tryptophan and leucine share a common transporter at the blood-brain barrier. LAT1, large amino transporter 1. It sits on both the luminal or blood and the abluminal or brain side of these brain microvascular endothelial cells, and allows for the transport from the blood to the brain. Leucine has a far greater affinity for this transporter than that of kynurenine and tryptophan, which means that if we hit a sweet spot with our dose, we can competitively inhibit kynurenine from binding to that LAT1 receptor and getting into the brain. The reason we discovered that we're not going to inhibit tryptophan from getting, block tryptophan from getting in is because LAT1 actually, the blood-brain barrier actually increases influx of tryptophan into the brain under conditions of inflammation. It actively tries to prevent tryptophan depletion, which is why we saw no reductions in the brain earlier. So we gave our mice leucine immediately prior to LPS and then 6 hours later to make sure it stayed in the system. Are you getting feedback on the little squeaks, or is it just me? No? Okay. I can hear it buzzing in my ear. So we kept them on leucine, we waited our 24 hours and we measured our depression-like phenotypes. Really exciting for us is not only did leucine block LPS-induced or inflammation-induced depression-like behaviour, we see sucrose preference maintained, we don't see the increases in the forced swim test. We were actually able to do this or show antidepressant properties of leucine in our control mice as well. Well, you'd expect then, if it's working the way that we think that it's working, that we would have a reduction in the brain levels of kynurenine. So we should be seeing less kynurenine in the brain in our LPS-treated mice. In the plasma, we see the increase in main effect of LPS, so it's doing what it's meant to do in the periphery. In the brain, we see a significant main effect in the kynurenine to tryptophan ratio, so in both our control and our LPS-treated mice. So how's it working? We thought it might be via LAT1, but we need to prove it. So we got in touch with Bill Banks over in Seattle, who is an expert in blood-brain barrier transport, and he used some Transwell. Well, not him, his student. Looked at a Transwell model where we can model blood-to-brain transport of whatever substrate you're interested in. You basically take the brain microvascular endothelial cells, place them in the Transwell, and you can load in radio-labeled tryptophan, leucine, or kynurenine, whatever you're interested in, and you measure the rate of transport from that luminal to the abluminal side. Now, the surface of the brain microvascular endothelial cells in the blood-brain barrier differ depending on if it's at the brain side or the blood side. So we can look at the influx transport to the direction of transport going into the brain versus going out of the brain by just flipping those cells over, right? Okay, we first looked at whether or not LPS or inflammation might increase LAT1. So this is some early QRT-PCR data where we see an increase in response to LPS in the heavy chain subunit of LAT1. We've now since shown, looking at protein, that you do get an increase in LAT1. And then we wanted to test whether or not leucine, tryptophan and kynurenine share the common influx transporter. So we went in and we added our labelled leucine, and this is the rate of transport, and we can see that when you then try and add in tryptophan, it competes with leucine and reduces the rate of flow across into the brain, or the brain chamber. Kynurenine does the same thing, and leucine inhibits itself. We also see that if we use a LAT1 specific antagonist, BCH. So the influx transporter between kynurenine and leucine is the same. We then wanted to test whether or not kynurenine can inhibit itself before going further. And you'll notice here that it certainly does, but the story gets complicated. What's happening here? When we just look at transport of kynurenine on its own, we're seeing it being pushed from that from the abluminal chamber, the brain chamber, back out into the blood side. So we've discovered a novel efflux transporter that predominates in the kynurenine system. So what this means is, as LAT1's letting kynurenine come into the brain, there's an efflux transporter working at a faster rate to try and push it back out again. The question is that efflux transporter also utilised by leucine? Because if we can use leucine to block kynurenine getting into the brain, great, but if it's also going to block kynurenine getting out of the brain, that's a problem. So we put in radio-labelled kynurenine and we see this effect again. This means that tryptophan shares that common efflux transporter with kynurenine, but leucine does not, and it's not LAT1 mediated. So to summarise, leucine, kynurenine and tryptophan enter the brain via LAT1, and we can therefore use leucine to block that entry into the brain, but leucine is not going to affect the predominant transporter for kynurenine, which is going to try to attempt any central excess levels of kynurenine getting back out of the brain. So we're quite excited about this. Okay, let's take a step back and come back to cancer. Okay. We know that peripheral tumours can induce cognitive impairment. Neuroinflammation, we believe, drives this effect. Anti-inflammatories may be able to prevent cancer brain, but we may need to target downstream mechanisms of neuroinflammation. The point for our little tangential episode was to say, hey, there are options, so the story's not over. We're chasing these down now in our mouse models of cancer. We've got IDO knockout mice sitting in the lab at home. In the last 5 minutes, I just want to switch gears and tell you a little bit about our model of chemobrain that we've gotten up and running. This is driven largely by a PhD student in the lab, Asen Chang, in which he treated mice with paclitaxel at a cumulative dose. So he gave 10 mg per kg IP every day, every second day, to get to a cumulative dose that looks equivalent to what a breast cancer patient would be receiving. He then did a battery of behavioural tests 24 hours after the end of that chemotherapy. He waited a while and did it again to see if there's persistent effects. Well, he found as well that we see this reduction in from even after 24 hours after the last dose of chemo in novel object recognition performance, and it's maintained into the second week. If we look using confidence interval indicators of what the incidence rate is of cognitive impairment in our chemo-treated mice. At around our age-matched healthy control mice sit at around 20%, whereas 50% or more of our mice treated with chemotherapy are showing some evidence of cognitive impairment. This looks awfully like the numbers you see in cancer patients receiving chemotherapy. He also saw an effect on sucrose preference, so this reduction in sucrose preference indicative of a depression-like phenotype, transient although it may be. We were worried that this isn't depression, but actually the mice can't taste anymore because chemo can really damage taste buds, and we see patients reporting a lack of taste sensation. So we went in and we counted the tongue papillae on the mice, and there was no difference there. It wasn't fun to do. But we also then wanted to check that the receptors weren't damaged. So when the gene TAS1R3 combines with 1R2, it allows for the perception of sweet taste. You'll notice that there's no change here. When TAS1R1 with TAS1R3 is combined, we have the perception of umami taste. Well, you'll notice that there's a reduction here in our paclitaxel-treated mice for the TAS1R1 receptor. This matches very nicely what cancer patients have also show, where they are encouraged to add in flavours that are going to boost their ability and sensitivity to umami taste. So our mice look like your patients, right? Just to prove to you that this isn't because we're causing chemo-induced sickness, no changes in body weight. Interestingly, we see an increase in burrowing activity, which is quite common to be commonly observed in mice. I think it's via glutamatergic processes, but it's just a hunch, so don't hold me to that. So chemotherapy induces changes in cognition and mood that are independent of sickness. The question is, how is it doing that? Well, there's two predominant hypotheses. One is this inflammation story again. The other is, no, chemo's getting into the brain and causing direct cytotoxic effects to those neuronal cells. Well, we looked at inflammation and we started off in the liver at some candidate genes using QRT-PCR, and we saw no evidence at 24 hours after that last dose of paclitaxel. But at 2 weeks, we started to see some markers come up. So we thought this was interesting, and we went and explored this a little further by using multiplex to check protein. And the story remains the same. We see very little, if anything, in regards to inflammatory markers at 24 hours post the end of the chemotherapy regimen. But at 2 weeks later, we start to see an inflammatory signal coming up. It's possible that inflammation is responsible for those late effects of chemotherapy, but it certainly doesn't explain what's happening at 24 hours. So we've started to investigate this by just checking whether or not paclitaxel can actually get into the brain. It does, and it does so in this model very, very well, despite what some pharmaceutical companies would have you believe. It gets in there. So we are currently chasing down what the hell is happening in these mice, and one way we're doing that is, of course, we're going to look at the central inflammatory profile to double check that. But we've teamed up with other researchers at Monash who are experts in non-targeted metabolomics. We're going to look at the metabolic pathways, all the metabolic pathways in the brain of chemotherapy-treated mice. It'll give us something that looks like this. Hopefully what we'll be able to do is find pathways that are changed between our chemo-treated mice and our non-chemo-treated mice using some bioinformatic approaches to allow us to then go in and start targeting and intervening at the level of those pathways to determine if they're involved in mechanism. Okay, 1 more minute. As I mentioned, we're moving towards this, trying to investigate if these multiple events of treatment, when taken together, are causing long-term changes that are more exacerbated or more prolonged than each individual event on its own. And at the moment, we're focusing largely at the individual events, but in the second half of this year, we're going to start that work. So we're getting somewhere. Now, I think you guys know this person predominantly. I'm in the Cancer and Neuroimmune Research Laboratory. What the hell am I doing in a cancer biologist laboratory, right? Well, our model is not only surviving but thriving. So, I think you're all familiar largely with what Erica's work does, so let's agree not to tell her that I'm going to summarise her career's work in about 10 seconds, okay? As you know, she's interested in neural signalling, or how that brain might talk to the body, and has shown that beta-adrenergic signalling or stress pathways interact with the tumour microenvironment and the tumour itself to enhance metastasis and cancer progression. My work looking in this other direction is quite complementary to that. So we've started to reframe how we're really looking at these brain-to-body communication pathways, and instead we know that when somebody has a tumour, I've hopefully convinced you today that we believe humorally that it talks to the brain and induces cognitive dysfunction. Now, these changes in behaviours are just readout measures in my mice for some brain damage, some issues there, and we believe it's priming the brain to be more susceptible to stress or to any insult, which is going to feed back and help those tumours grow. This may not be a coincidence. And what's most if we see this as a positive feedback loop between the brain and the tumour, we may be able to use the same interventions, and of course we want to look at treatment, how treatment intersects that as well. But we may be able to look at the same interventions that can actually target cancer progression as well as cancer and cancer treatment-induced side effects, which is where we're heading. Okay, I'll leave it there. Thank you. Sure.
So I'm wondering if epidemiologically, is there evidence that women who have been on low-dose aspirin prior to their breast cancer diagnosis fall into the resilient category at a greater rate than the women who fall into the chronic?
Right, so there's definitely evidence showing that aspirin is protective. There's currently a large-scale...
Protective of cancer development or protective of the symptoms after?
Oh, I'm sorry. Were you asking about the symptoms?
I'm asking about the symptoms.
Yeah. Oh, so sorry.
It's breast cancer. With what your mouse model suggests with the treatment with aspirin, that it could prevent having this response. Right.
I'm not familiar with any research that has shown it. There is a large. So the reason that we jumped onto aspirin to begin with was to provide some evidence to try and join a large study back home called the ASPRI trial, and it's looking at the benefits of aspirin in multiple diseases, including dementia and including cancer, but they're not looking at cancer-induced cognitive function. So hopefully we'll be able to do it, but I'm not aware of any research that's shown it. Probably anti-inflammatory other factors, there might be some out there, but nothing that's specifically focused on it epidemiologically. Sure.
Thank you very much. That was very interesting. I was wondering, you showed in your model that there's a vulnerable subgroup, and that's what the literature seems to show. In your mice model, what types of pre-existing vulnerabilities do you look at, or who might be more vulnerable to the effects of inflammation, the effects of chemotherapy, the persistent cognitive effects?
Right, so we haven't done that, but if you're interested in what happens before you get cancer and what pushes you there, so my PhD was in early life inflammation and looking at long-term susceptibility to anxiety and things like that. So we certainly have experience and could go into looking at early life stress models and so forth. We're very interested in, I didn't talk about the sleep work, but looking at modelling shift workers who are on an adjusted light cycle, get poor sleep, circadian rhythm disturbance, and then start developing tumours. That's probably where we're going to go first. But yeah, we can, any behavioural intervention or any insult prior to the tumour, it's possible. Any model that you see in mice. Yeah.
I have a basic and methodological question. Whenever I see animal studies, I wonder about the translation to humans. simple basic question for those of drug medication and biologic using animal models and then human models. You mentioned that the aspirin amount you gave mice were all like low dose 81 or 30 of aspirin in humans. So how do you work converting doses between animal model and human model?
Yeah, very difficult.
Yes, using all these drugs.
Very difficultly. It depends on how the mouse compares in terms of metabolism of whatever substance or compound you're trying to inject. The aspirin data came from a study where we checked the amount of mice that the amount of water and aspirin mice typically will drink per day and work from there. And in doing that, there's been a published pharmaceutical paper that's then shown, based on those calculations, that it sits at around 100 milligram for humans. There are conversion rates based on meters squared, so if you're doing chemotherapy from humans to mice, but it's a model. That's why you have to then test it in the clinic. Yes?
Yes, in your test model, mouse model, you use the 41% mouse.
Yes.
Other that I can use similar types of
So that's what we're doing. So, full disclosure, we picked 4T1 to start with because it is rather inflammatory. We've got a metastatic breast cancer model in C57 black 6 mice that we're going to be testing, or hopefully is getting tested right now, unless the students have decided to take a break because no one's there. So we will be doing that. The other thing that we're particularly interested in doing is using, so that answers if it compares with other breast cancer cell lines. Is this a generalizable effect to any solid tumour of the periphery? So we're going to be using a mouse model of colorectal cancer to determine if that too, so in a completely distinct anatomical site, if that too also causes cognitive impairment. And then look at, is there any overlap between the metabolic or the inflammatory signatures? Do we have a commonality of cytokine mechanism? So not yet, but we're doing it. Sure.
Thank you for a great talk. Very interesting. I had a question regarding the assessment of cognitive function. If there were any other measures in the mice outside of the short-term memory?
So yeah, that's something that we would love to explore further using, there's some iPad tests with mice that you can really get in and do some more sophisticated analyses. We have looked at it in terms of spatial learning and spatial memory as well, and we don't see any changes there. So if one is equal to the other, it would tell us that maybe in our novel object recognition task, it's not about whether they learn the test, it's more about memory. But yeah, we haven't done too much.
And the spatial learning and spatial memory wasn't changed?
Correct.
At that initial phase, do you see it in long-term effects of the chemotherapy?
Oh, sorry, that was in response to the tumour. In response to the yeah, that was in response to the tumour. We haven't done it yet in chemotherapy. Sure.
Great talk, thank you. In the first part of the talk where you described the simplified sort of horizontal model, you asked the question, what are the mechanisms linking the tumor to the cognitive changes? And so you implicate inflammation, and you mentioned that you treated the mice with tumor-derived factors. Were those cytokines? I might have missed that part.
Sure. So it was the tumor cell conditioned media. So anything that the tumor cells have spat out into that media was injected. So you're absolutely right. It wasn't just cytokines. It wasn't just chemokines. That's why we need to mechanistically go in and block those cytokines and chemokines to make sure that that's the effect that's happening.
From the other work, you infer that it's cytokines that are part of that mechanistic pathway, but not just from that.
Exactly, yes. Yeah. We've got a lot of supporting evidence. We need to nail down mechanism.
Okay, so let me just ask a follow-up to that then. If it is the cytokines in the periphery that are driving that, then how do you know specifically that it's cytokines from the tumor versus cytokines from any other factors, including, innate immune system activation as a function of stress and so on and so forth? So I would say that cytokines could be involved, but that cytokines from the source of the tumor may be only one.
Yeah, so perhaps I wasn't clear. You're absolutely right. That peripheral inflammation, no matter what. So I would suggest that the tumours are going to be releasing or initiating a peripheral immune response and inflammation, but whether or not it's predominated by coming from the tumour or coming from the periphery itself, we don't know, and I suspect both.
Okay.
So your leucine data are really provocative, and I know it's kind of outside the purview of your talk, but I'm sure that you're aware of the literature. Can you talk a little bit about the translational studies that have been done with leucine, and how they've been done?
So they are being done. So pretty much the only work that's been done with leucine in patients has been around its ability to build muscle mass and fight cachexia. Robert is actually working with, involved in now getting some clinical studies to test leucine's ability to block inflammation-induced depression. I was talking to him briefly at the conference about that. So that's underway. Yeah. I've sort of left
So there's no
since.
studies that you're aware of that have taken depressed patients that may have inflammatory activation with high CRP or whatever you find and treated those patients?
With leucine?
Yeah.
Not that I'm aware of.
And you talked about that flux transport of kynurenine.
Mm-hmm.
And I didn't get what clinical importance that be regarding that mechanism. Understood the leucine part.
So in terms of understanding how or where leucine is having its beneficial effect at the blood-brain barrier?
So it's a LAT, as you said.
Mm-hmm.
And I think here, if you were thinking already about some other mechanism of blocking or using that efflux transport system to influence depression behavior.
So, sure. So we're interested in identifying that efflux transporter. It is much harder than it sounds. So that we could capitalise on that to also flush the brain of kynurenine. In fact, what I'm most interested in, or where I'm going to follow this up, is actually doing something called glutamate scavenging. So I talked about you have excess glutamate that's then in combination with that quinolinic acid causing the depression effects. In stroke patients and patients with traumatic brain injury, they're testing whether or not if you administer oxaloacetate to patients, it metabolizes all of the peripheral glutamate and causes which then causes, what's the word I'm looking for? Transport of that glutamate, that's extra glutamate in the brain, out to the periphery. So we might be able to suck the extra glutamate from the brain, and therefore another avenue for treatment.
I guess you have some data regarding sleep and circadian rhythm, but you didn't talk about it today. So do you find any relationship between sleep and circadian rhythm and kynurenine?
Okay, so we haven't done much on the sleep and circadian rhythms. I know my colleague who I'm working with that has shown relationships, as many people have, between sleep and inflammation. We haven't looked in regards to sleep and kynurenine, and I don't think anybody has, but I love that idea.
Just to follow up on the transport question, if you block the transporter, won't you be blocking transportation of other factors as well? Actually, may cause additional complications from that.
That's why we don't want to block it. That's why we're trying to competitively inhibit it, so some still gets in. But yeah, you're exactly right.
Thank you very much, Adam. Really.
Okay, thank you.